The citric acid cycle. The citric acid cycle and the transition phase are usually combined together throughout the process of cellular respiration. The only way the citric acid cycle can occur is if oxygen is present. With pyruvate being produced in the cytosol, it will go through the process of fermentation if oxygen is lacking. If oxygen is present, the oxygen will be found inside the matrix of the mitochondrion. What it will do is actually pull in the pyruvate from the outside of the cell of the mitochondrion into the mitochondrion because it is so electronegative. And because of this electronegativity and its desire for those electrons, it will allow for the oxidation of our glucose to continue. Glycolysis is just a partial breakdown of glucose. It is simply taking an organic molecule that is 6 carbons in length to a 3-carbon organic molecule in the form of pyruvate. But glucose is not oxidized in this process. In order for it to be completely oxidized, we need to strip it of all of its hydrogens and electrons in those chemical bonds. So the process of the citric acid cycle allows for this to occur. It is extremely important to remember that it is not going to happen unless oxygen is present. If we look at the mitochondrion, just a little detail again, we can see that it has this outer membrane. It has a space actually between the outer membrane and the inner membrane. If here's the outer membrane, here's the inner membrane. There's this space in between. The inner membrane is actually in all of these tremendous folds, which helps to increase the surface area of the mitochondrion. It is along these folds that we see the third process, or this electron transport chain of cellular respiration actually occurring. Inside these cristae, or these folds, is what we call the matrix. It is in the matrix where the citric acid cycle actually occurs. It is also in the matrix where we find the DNA, the mitochondrial DNA, and ribosomes. So a brief overview of what's going on again is we have glucose that goes through glycolysis. It gives us a net production of 2 ATPs. We produce 2 pyruvates in the process. If oxygen is not present, the pyruvates will actually go through 2 different forms of fermentation. If oxygen is present, then we will continue on into the citric acid cycle in the matrix of the mitochondrion. The transition step of glucose occurs when we transport the pyruvate across the outer membrane, the intermembrane space, and the inner membrane of the mitochondrion. These steps is a nice little process of reactions. We have here pyruvate. We have oxygen inside saying, hey, bring me those hydrogens and electrons into the mitochondrion, into this matrix. Well, in order for that to happen, pyruvate must go across both of these membranes. As it goes across the membrane, we are going to see a carbon dioxide being released. This COO with a negative charge, the electron hanging on, will actually allow for carbon dioxide to be released. This is the first oxidized carbon molecule. As we are oxidizing this carbon dioxide, we are actually going to reduce NADH. So NAD plus is going to become NADH plus H plus. If this is removed, all that's left is this 2-carbon sugar known as acetyl or acetyl. As we bring it in, we release the CO2, we reduce NADH, and then we attach a coenzyme to the acetyl and sugar and create acetyl-CoA, or coenzyme A. This is somewhat of an unstable bond, but we have this reaction occurring here. This is the transition step. This is how we get pyruvate across the membrane of the mitochondrion. What you need to be aware of is that this reaction has to happen twice because we have two pyruvate molecules involved at the end of glycolysis. So both pyruvates will enter or go across this membrane. As they go across, that means there will be two CO2s, two NADHs, and two acetyl-CoAs produced. This reaction is the forerunner of the citric acid cycle. Once we have our acetyl-CoA, we can bring that into the citric acid cycle. This is truly a cycle. The products create the reactants of the next reaction, and the final products are the initial reactants. To summarize the overall process, we see per pyruvate, per one turn of the citric acid cycle, 2 more carbon dioxides being released, 3 NADHs being reduced, 1 FADH2 being reduced, and the production of ATP through substrate level phosphorylation. Because this has to happen 2 turns or 2 times, we end up with a net gain of 4 CO2s, 6 NADHs, 2 ATPs, and 2 FADH2s. If you recall, the whole purpose of cellular respiration is to completely oxidize glucose. By the second turn of the citric acid cycle, glucose is completely oxidized. We have 1 carbon dioxide, 2 carbon dioxides, 3 carbon dioxides oxidized per pyruvate. We need 2 pyruvates, which will end up being 6 carbon dioxides. If you recall your formula, the first product that we see in our formula is 6 carbon dioxides. By the end of the citric acid cycle's second turn, we have completely oxidized glucose. The majority of the energy, however, is stored or trapped in the NADHs and FADH2s. Because recall, glycolysis only gave us a net gain of 2 ATPs. The citric acid cycle will only give us a net gain of 2 ATPs as well. That is only 4 ATPs and not enough ATPs to generate all of the cellular work that we require. So let's look at the citric acid cycle in a little bit of detail as well. We start with acetyl coenzyme A and oxaloacetate. Oxaloacetate is a 1, 2, 3, 4 carbon sugar. Acetyl-CoA is a 2 carbon sugar. When they combine together, we create a 6 carbon sugar known as citrate. This is where, or citric acid. This is where we get the name, the citric acid cycle. Oxaloacetate, acetyl-CoA come together and form citrate, a 6 carbon sugar. Citrate then will go through an isomerase enzyme and create isocitrate. From there we take isocitrate, and here we are actually going to reduce our first NADH and release a carbon dioxide. And as you look at this, you can see that the hydrogen here and the hydrogen here have been removed to form the NADH. And we also have removed a carbon dioxide right here. We now have a 5 carbon sugar alpha ketoglutarate remaining. Alpha ketoglutarate again will go through and oxidize off a carbon dioxide and reduce NADH plus H plus again, and leaves us with succinyl coenzyme A. So by the point of step 4 of the citric acid cycle, we have completely oxidized our first pyruvate molecule. We then see a series of reactions occurring all through substrate level phosphorylation to produce one ATP molecule. Succinyl-CoA then becomes succinate, which will then reduce another FAD or a FAD molecule to FADH2 to give us fumarate. And if you're following, you could see here's a hydrogen and here's a hydrogen. Two hydrogens, CH2, CH2, now it's CH and CH. Those other two hydrogens are right here, as well as their electrons. We go from fumarate to malate, which is just a rearrangement of those chemical bonds, and then malate is reduced or allows for another NADH to be reduced and gives us oxaloacetate as our final product. But remember, our final product becomes the reactant of the next cycle, and we go through this whole process a second time. Here is another way of looking at the citric acid cycle. Per pyruvate, and that's the key, per pyruvate, we take this pyruvate molecule across the membranes of the mitochondrion. As we come across the membranes, we are reducing an NADH, we are releasing a CO2. That's one, that's two. We now have acetyl-CoA. This goes into the citric acid cycle, formerly known as the Krebs cycle, so if you come across that, don't be alarmed. It is the same thing. The Krebs cycle. Through the process of the turns of the Krebs cycle, we reduce 3 more NADHs, 1 ATP is phosphorylated, and 2 FADH2s are reduced. We also have 2 carbon dioxides here on the end. 1, 2, 3 carbons to pyruvate, 1, 2, 3 carbon dioxides released. Pyruvate is now completely oxidized to the inorganic molecule of carbon dioxide. Where is the energy that was stored in all of these sugars? Right here in the NADHs and FADH2s. Complete oxidation. Now let's watch a video that summarizes these two reactions as well. Okay, as we begin, we have our pyruvate molecule in the cytosol that was produced through the process of glycolysis. The first reaction that happens is our transition step, taking this pyruvate across the 2 membranes of the mitochondrion and forming acetyl coenzyme A. So what we will see happening is this pyruvate going through, NAD plus being reduced to NADH, and carbon dioxide being oxidized, and the coenzyme A attaching itself. We now take coenzyme A into the citric acid cycle by creating a 6-carbon sugar with oxaloacetate. The 6-carbon sugar is citrate. The citrate then almost immediately goes through a reaction and forms its isomer, isocitrate. It's just a structural isomer. From here, the 2 carbons that were added to oxaloacetate will slowly be oxidized off. NADH reduced, CO2 removed. NADH reduced, CO2 removed. ATP produced through substrate level phosphorylation. FADH reduced to FADH2. NAD plus reduced to NADH. So at the conclusion, or at the end of our cycle, per pyruvate, we have 3 carbon dioxides, 4 NADHs, 1 FADH2, and 1 ATP. This entire process allows for all of these electrons to be completely removed from the carbon dioxide, or from the pyruvate. The only reason these electrons are being removed is because of oxygen being there in the matrix, drawing them towards itself. Remember, oxygen is the greatest oxidizing agent, meaning it's so electronegative it will pull these electrons all the way downhill towards it. As it pulls them downhill towards itself, we end up seeing the production of 3 ATPs for every 1 NADH. This would be a tremendous waste of energy if oxygen was not there to convert it. And so we do not see the citric acid cycle happening if oxygen is not present. So if we look at this process one more time, we start with acetyl-CoA formation. We then form citrate. And then we see the reduction of several more NAD pluses and oxidation of 2 more CO2s. ATP made through substrate level phosphorylation, FADH2 reduced, and to get back to oxaloacetate, 1 more NADH reduced.